Okay, in the last part of the lecture, we talked about the levels of protein structure and the interactions that were involved in each. So we know that there's 4 levels of protein structure. Primary is the amino acid sequence, and we have the covalent peptide bonds, which are also known as amide bonds. Putting those together. We have secondary structure, which are only a local 3-dimensional structure, meaning not the whole story. And those are alpha sheets and alpha helix and beta sheets. And those are interactions in the backbone. Hydrogen bonding between the hydrogen that is covalently bound to the nitrogen and the oxygen on the carbonyl, bless you. And so those are the 2 structures that are involved in the secondary structure. Tertiary structure, we have the involvement of the R group, and we have some non-covalent interactions between those. So we have our hydrogen bonding, hydrophobic interactions, electrostatic, meaning charge attractions and repulsions. And then we have also one covalent interaction called the disulfide bond between 2 cysteine amino acid residues. The last level is quaternary. The quaternary structure is when you have more than one subunit, and those subunits come together and interact in the same way that the tertiary structure interacts. So you have R group involvement. We ended up talking about how we determine the tertiary structure. We can try to predict how a protein is going to fold based on its amino acid sequence, but it's not always going to be accurate. So we'll have to rely on either X-ray crystallography or in more modern times, and a much more complex technique is the 2D NMR. Myoglobin was the first protein for which X-ray crystallography was used to determine the structure. It is a great example, very simple example of a tertiary structure. And many of you know the function, know a lot about myoglobin, but can you tell me what the function of myoglobin is? Stores oxygen where? In the muscle, that's right. It's only a single subunit, pretty short, 153 amino acids. And it has what we call a prosthetic group. So let me talk about what that means. A prosthetic group is just like a prosthesis on a person. It's something that's non-human that you add to a human to make them whole, right? To make them functional. So here we have a non-protein heme group. The heme group is our prosthetic group that attaches onto our protein to make it whole. We're going to talk a lot about a heme group later on, but really what the heme group is the part that makes the myoglobin active, and it does the binding of the oxygen. A lot of aqueous environment proteins have a lot of alpha helix. This one does not have any beta sheets, and again, the polar side chains are on the surface to interact with water, and nonpolar side chains, the R groups, are folded into the interior. Now, at the site where the heme group is involved, we have 2 histidine side chains, and they're involved with the interaction with the heme group and the oxygen. So the heme group has what we have coordination sites. We're going to talk about that in a moment, what does to coordinate mean. But the 6 sites, 4 interact with nitrogen atoms on the heme, 1 with a nitrogen on histidine, and 1 with either an oxygen molecule or on a nitrogen on another, what we call gate histidine. So here's the structure of myoglobin. You can see in the red we have where the heme group is. And then the green are just the amino acids. And I want you to notice that scientists have named the regions of the of the myoglobin structure. So here's the N-terminus and here's the C-terminus. And we might say, oh, there's a glycine in the GH region. And so people will know what you're talking about. So they can kind of visualize. People that spend their entire lives thinking about myoglobin, they'll and there's people that do. My research advisor spends her entire life thinking about tubulin. So, there's people that spend their lives thinking about one single protein. So you can see that we've labeled them letters. So just be familiar. You don't have to be familiar with this one, but just know that that's what that means. These letters is just so that people have a little shortcut. Oh, we're in the FG region here, so that they can do that. Let's talk about this heme group. This heme group is iron 2 with 4 5-membered rings, and they're called protoporphyrins. They're pyro-like rings. You guys that haven't taken organic don't need to know that, but just know that they're ring structures around. The blue are nitrogens. So the blue ones here are nitrogens. And let's talk about what it means to coordination chemistry, or to covalently coordinate with the iron. It's hard to say. Iron is in the transition metals, right? And what orbitals do transition metals have access to that other metals in, say, group 1 and 2 do not, in the upper part of the periodic table? Come on, chemist. One chemist in here. D orbitals, right? D orbitals, right? And there's some empty d orbitals. So what a coordination bond is when both electrons come from the nitrogen and rest in those empty d orbitals. So there's a lone pair on each one of these nitrogens. And the lone pair is resting inside an empty d orbital on the iron. So here, these silver bonds here, the electrons aren't shared. They're not coming from the iron. The iron still has a 2+ charge. But what it is they're just residing in, they're like freeloading in the empty d orbitals. And so here, this part down here, this comes from the myoglobin. So this is sort of like an anchor. And this coordinate covalent bond stays there. On this histidine E7, this one sometimes coordinates with the iron, but when oxygen's there, it doesn't. So what I like to think is this is the coordinate covalent bond are like fangs that kind of hook on to our iron and hold it in there, hold it in place. So the fangs come from the snake, not from whatever the snake is going into, right? So the nitrogens have the fangs, and it kind of just grabs onto that iron. So I do expect you to know what a coordinate covalent bond, because we're going to have a lot of coordination going on. Metal ions are important. So this iron, when it's not coordinating with this histidine, will coordinate with oxygen. Oxygen has a lot of lone pairs, so the oxygen will come down and coordinate with it. When it doesn't have oxygen, it will go up with this histidine. So this histidine E7 is sort of like a gate. Now, only when myoglobin has this nice structure, is folded in this nice way, on this particular way, is it native. And only when it has the heme is it active. But some conditions cause denaturation, where we're losing our quaternary or our tertiary and sometimes our secondary structure that will give the loss of biological activity. One thing I want to make sure is that you understand that we are not degrading the protein. Degrading means that we lose the primary structure. We still have the peptide chain intact. When we denature, we still have the peptide chain intact, but we're losing the biological activity. So we lose the biological activity by losing the quaternary structure if it has it, the tertiary structure, and then sometimes the alpha helix and beta sheets will also get lost, but not always. But by losing that tertiary structure, you're going to lose your biological activity. Sometimes we want to denature, sometimes we don't. When I was in industry, I would like to hire chemists, but the problem with chemists is that they would not know how to work with proteins, because some proteins, if you look at them wrong, they will denature. I would like to hire biologists because they would know how to work with proteins, but then they wouldn't know the chemistry that was needed. So it was very difficult to find the right person to work in my lab. So I would just have to teach. And then I realized, I really like to teach. And I came here. So see how life works. So one thing that for sure causes denaturation is heat. You guys are right? Can I answer a question? Is it? No. Okay. Interrupt me if you guys are behind on something or need a little extra clarification. I welcome it. It slows me down, so that would be a good thing. So heat. Let's think about, what is heat doing? When you heat something up, what are you doing to the molecule? You're causing an increase in what kind of energy? Kinetic or movement, right? If you're moving, what can we do? We might break those intermolecular forces that are holding it together. So all those intermolecular forces that we listed for the tertiary structure could be broken. Maybe not the disulfide bonds, but you might break the other ones. So heat causes vibration. So I will need you guys to know not only the definition of denaturation, but also what causes denaturation and why. So again, the definition of denaturation is the loss of biological activity through the loss of our structure. We're not missing that primary structure. Primary structure is still intact. If we lose that primary structure, it's called degradation. So heat causes vibration, which breaks our intermolecular forces. Large changes in pH. Remember, if we're changing pH, we can change the charge on our acidic or basic side chains. So if we are having a negatively charged carboxyl group at neutral pH when we're above the pKa, if we go really low, we might protonate it. And so it's uncharged. This is supposed to be NH3, and this is supposed to be NH2. Sorry for the question marks. I know them. So here at physiological pH, this is going to be NH3 with a positive charge. But if we go very high in pH, this is going to be NH2. So what kind of interaction would large changes in pH cause? What attraction or repulsion? I call them what kind of bridges? Salt bridges. So large changes in pH can affect our salt bridges. Can also affect our electrostatic repulsions. The next one is soap. SDS is soap. It's a detergent. Have any of you, I may have asked this question, done SDS gel electrophoresis? Maybe? No? Some of you? When you must have seen them on TV, at least, where they have the gel and the blue bands on that. So in that technique, what we are doing is we're separating based on size. Now, if we have the protein and it's folded up, fetal sort of position, all folded up nice in a nice little tight ball, then we're not really separating just on size, we're separating on shape. So when we want to separate just on size, we unfold the protein and then run it through this mesh of gel. And the big proteins get caught up early in the gel, and the small ones can travel further down. And then you stain it with something that stains to proteins, and you see those little nice little bands. Some of you must have. You've done it, okay. And so if you've done the non-native one, the SDS gels, what you're doing is you're adding soap so that just like you get soap out of your soap to use to get the grease off your hand, what we're doing is SDS has a long tail and a polar head. That long tail finds its way into the hydrophobic parts of the protein. And then it makes that polar side of it. So it makes the non-polar parts of a protein polar, so it unfolds. Remember I said that hydrophobic interactions are the main driving force for the folding of proteins. So by adding soap, what we're doing is that hydrophobic tail makes its way into the interior of our protein and has that polar head on it. Does anybody need any further explanation on that? So then just like if I had a little piece of grease on my hand and I put soap on it, soap has a long hydrophobic tail, hydrocarbon tail, that dissolves itself into the grease on my hand. And then what we see on the surface of the grease is just the charged part of the soap. And then so the water can wash it away. And that's the same thing with the detergent, such as SDS. So normally we want this to happen when we're doing this technique, so detergents will disrupt the protein. Urea or guanidine is really a neat thing. Urea, guanidine, we call them chaotropic agents, C-H-A-O tropic. I like to think of it as chaos causing. And these chaotropic agents disrupt hydrogen bonding. These molecules, what they can do is do a lot of hydrogen bonding. So CHAO tropic. So it's CHAO tropic. And so they will hydrogen bond. They'll go around and hydrogen bond in the place of things that should be hydrogen bonding with other things. So they put themselves in to where maybe two polar amino acid side chains should be hydrogen bonding with each other. These guys go in there and disrupt that hydrogen bonding. And in the old days, running a gel used to be a smelly job because we would not only want to denature our protein, but we would also, in order to completely denature, we would add not only SDS, but mercaptoethanol. And what that does is it reduces a disulfide bond. And this stuff is smelly. I think they have new chemicals that aren't so smelly because it was really a smelly job. So this is modern times. No one gets a perm anymore, right? Are perms coming back at all? No. That's just too much work. In the 80s, it was high maintenance. And both men and women would get perms. So what a smelly, and that was pretty smelly, because what they would do is you'd put the curler in your hair, you'd break all of the disulfide bonds, and then you'd add then you would rinse, and then you'd add another chemical that would oxidize them, and so that it would form the disulfide bonds in the shape that your hair was in the curler. It was not nice. I'm glad perms have gone away. That was...
Now they do the opposite. They do straightening ones.
They do straightening ones.
Yeah. Yeah, now straight hair, so like they'll flatten your hair and put it on.
Wow. So they have like a flattening type of process? Is it chemical?
Straight hair. Yeah,
the
like
keratin. The keratin treatment is
The keratin is
it's an actual perm.
It's an actual perm, but it's...
Instead of putting your hair in rollers, they lay it flat.
Oh, so they break the disulfide.
If you have curly hair, you'll wash out with straight hair.
Wow. That's just a lot of work. I am ashamed to say that in the 80s, I had perms.
Oh my God.
My mom.
Do you have pictures?
No. No pictures.
Please.
I
Have ways.
had very big hair. I was from north of Boston, and it's like that area just, the bigger the hair you could have, the better. Yeah, okay. So this is a nice little picture of what's happening. Here's our active, correctly folded native protein, and here's our denatured protein. But look, there's a double arrow. What does that mean? You can go back. We can refold if you're nice. Okay? There's some points that there's no going back, but you can refold it. And I can tell you here what we're doing here is adding to say this is ribonuclease. What we're adding is urea and beta-mercaptoethanol, and here we get the denatured reduced protein. And then what we do is sort of do dialysis, and we allow, you remove the urea through dialysis, and then you allow it to air oxidize, and it will pop back in. Now, this only works for small proteins. You have a large one, it's going to be really hard to do this. Not to say that it can't. Yes, Patrick?
Something like a prion being refolded?
A prion causes other proteins to not fold correctly, so it interferes.
How it would interact if it would stop it, or it would...
So a prion, I don't even know what a prion really is, but a prion causes other proteins not to fold correctly.
So any enzyme that fit would fit.
So when we talk about folding later on, there's something called chaperone proteins that aid in the correct folding of proteins. And so I think it has something to do with that, where it's causing those proteins to not work properly and not have the... So one of the first biotech drugs that I worked on was made in E. coli, all right, and it was made in the in what we called inclusion bodies. So we had a vector, DNA vector, that we put into the E. coli, and it made this small protein. We called it axokine, and it junked up this like glob inside the bacteria, and so it was an insoluble glob. So what we did is we had this procedure to break open the E. coli and spit out what we called inclusion bodies. So we would grow a lot of E. coli, and then have this process by where we would isolate the inclusion bodies. So at that time, what I would have is this solid mess of protein. But our job was to get it into the aqueous environment so we could purify all of the stuff from the E. coli out. So what we did is we took the solid and we put a bunch of urea in it. And what that did is it got it to be solubilized. And then we had to slowly remove it from the remove the urea from the solution. And because this axokine was a small protein, it would pop back in. This was part of the manufacturing process by which we made that drug. The drug never made it to market, although drugs have since made it to market from Regeneron, but that was supposed to be an this one was a weight loss drug. It started out as a treatment or its predecessor started out as a treatment for Lou Gehrig's disease. And what happened during the clinical trials is that people started feeling better with Lou Gehrig's disease, but they still died. And what they realized is they started feeling better because they lost weight. They got more mobility because they lost weight. And we said, this is our little blue pill. But no, it wasn't. So I remember being in the executive suite when the phase 2 clinical trials came out. And it was a 2 or 3 month study, and people were losing weight. And the guy that ran the clinical trial was saying, there's stories of participants that they're buying better food. Their families are angry with them because they're not buying the junk at the grocery store. I thought that was interesting, that their families would try to sabotage them losing weight. And I guess that happens. I'm sure that happens when, one person tries to get healthy and the other people aren't healthy. There's probably a lot of bad feelings. So it was very interesting. But then we went and did the largest phase 3 clinical trial that had ever been done at that time. And they found out that right after that phase 2 ended, if they had extended it one more month, they would have found that they stopped losing weight and started developing antibodies to the drug, which is a big no-no. You do not want to develop antibodies. You don't want to change a person's immune system by giving them a drug. You can't. The FDA does not look kindly on that. So we cried. But then they were happy afterwards, and all the stock went up to 500. It's down now. But, yeah, you guys want a job in the biotech industry? We had a meeting here yesterday with Regeneron. Did you go to that? Was it good?
Yeah, it was good.
So, yeah. It's a great company to work for, and I can speak from experience. It was very hard for me to leave there. And they're just growing like gangbusters. So I know I had a neighbor that, when I was still there, had connections. And I got him to have a summer job before he went to med school. And I believe it helped him get into med school by doing a summer internship there for a couple of years. So just because you might not want to go into the biotech field, it's good to do stuff outside of your comfort zone. I think med schools look for that. All right, quaternary structure. It's not as common. It's where two or more protein chains or peptide chains come together to form our biologically active unit. So they are subunits, those protein chains. And if all the subunits are identical, that's called an oligomer. If it's two subunits, it's called a dimer. That's what my tubulin was, a dimer. Not as common as trimers and tetramers, but we know hemoglobin is a tetramer. It has 4 subunits that come together. And again, the same interactions that put together our tertiary structure are going to put together our quaternary structure, our subunits. I believe that you can have your disulfide bonds as well. Yes.
Did you say the one was a ligamer?
A ligamer, if it's two of the same subunits coming together, that's a ligamer.
Okay.
Okay, so proteins with quaternary structure. There must be some reason why nature has put subunits together. Why didn't they just make it one big chain? Well, it's because they often can be what we call allosteric, where one subunit will bind to a molecule, and that molecule that it binds to is called a ligand, and cause a change in the structure. We see a conformational change in other subunits that makes it either more receptive or less receptive to a molecule. And we see this kind of behavior in hemoglobin. A little bit about hemoglobin first. We know it's to transport oxygen where? In the blood, right? It's a tetramer, and it has 2 different types of chains, 2 alpha chains and 2 beta chains. This is supposed to be alpha and beta. And each of them, whether it be alpha or beta, has a heme group. So there's 4 heme groups. Each one can bind a molecule of oxygen. And what we see is positive cooperativity. When one oxygen binds to one of the subunits, it becomes easier to bind other oxygens. There's a conformational change. Once one oxygen comes in and binds to one of the subunits, that changes the conformation of the other subunits, and it makes it easier for the next oxygen to bind. That means that the structure of hemoglobin, HB is for hemoglobin, without oxygen is different than the structure with oxygen. There's other molecules that can affect the ability of hemoglobin to bind oxygen. And we're going to talk about the protons, carbon dioxide, and BPG in a moment. Everybody good? Any questions so far? All right. So here's our hemoglobin. I get to have the nice colors. You guys have not such nice colors. So the blue are the alpha, and the green are the beta subunits. And you can see each one has a heme. All right, we're going to spend some time on the next few slides. But the binding of hemoglobin for oxygen is decreased by the presence of hydrogen ions. Protons cause it to bind poorly. Also, carbon dioxide cause it to bind oxygen poorly. And BPG also makes oxygen bind to hemoglobin poorly. BPG is a very interesting molecule. It is there to make sure that we get rid of every last bit of oxygen on our hemoglobin. So you see this molecule in our blood, and what it will do is it will basically kick off any oxygen. Pregnant women have a 30% increase in BPG over the rest of us. You don't need to know what these C1, 2, and 3. Don't worry about those. And interestingly, fetal hemoglobin binds BPG to a lesser extent because it's going to get it. So the mom has a lot of BPG around. The BPG is going to come in and kick out any oxygen that's on the hemoglobin. The oxygen will be floating around, go through to the baby. The BPG will not affect the binding of oxygen to the baby hemoglobin. So that's how oxygen gets from mom to baby. Because if they had both the same affinity for oxygen, there'd be no reason for it to go from the mom to the baby, right? Babies take whatever they want. They will drain your bones of calcium and of every nutrient and every last bit of energy. And you're left with nothing. And they come out nice and healthy. Evolution at its best, right? It leaves the mom ragged and come out with healthy babies. I had very heavy babies. All right, so let's take a look at this. What happens? So we have this like axis in the middle between our alpha and beta, some sort of symmetry there. And when we bind, we have this turning of 15 degrees of this axis from the one alpha beta section to the other alpha beta section. So it's a turning. They slide by each other. And that's because salt bridges and certain hydrogen bonds are ruptured. But next what we're going to do is talk about how pH affects the binding of oxygen to hemoglobin and how it's different than the binding to myoglobin. So there's two concepts that we're going to cover. We have a very busy slide. Next slide. So what we say is myoglobin binds to oxygen normally, while hemoglobin shows what we call cooperative behavior. So what we mean by cooperative behavior is that each oxygen added to the heme makes it easier for the other ones. Myoglobin only has one heme, so there's nothing else going on. It binds to oxygen and it lets the oxygen go. It doesn't really do much. But because hemoglobin has 4 subunits, each with its own heme, it has an effect. Once you bind the first one, it's going to affect the other one. So we see this in the binding curves in their shape. And this is really important, guys. If I start talking about something and you're not getting it, you need to be vocal right now so that I can have you with me. So myoglobin curve is what we say hyperbolic, which I'll get to in a second. And the hemoglobin curve is sigmoidal. So again, I said this slide is very, very busy. So we're going to spend some time on this. Have you guys seen this before? This slide, the oxygen binding slide, or a curve? No? Yeah? So in other class, what class did you see it in?
Bio 2.
Bio 2?
Paramedic class. So.
Okay. So we see this shape as hyperbolic. All right? This myoglobin, that shape I want you to recognize is hyperbolic. And the hemoglobin shape is what we call sigmoidal. Looks like an S. That's easy for me to remember. I remember myoglobin is hyperbolic because it's hyper. It goes up quick. Right? That's how I'm going to remember that. And then the sigmoidal shape comes from it looking like an S. All right, I want you to just take a look at the myoglobin. On the y-axis, we have the saturation with oxygen. What does that mean? Here, this 0.5 means that half of the myoglobin molecules, proteins, are with oxygen. This 1 means all of them are. So that's what the y-axis means here. And the x-axis is just concentration of oxygen. So we're going more and more oxygen on the x-axis. So let's spend some time with just the myoglobin. We can see that it doesn't take very much for our myoglobin, very much oxygen for our myoglobin to get filled with our oxygen. But as we go up and up, what's happening? It's harder and harder, right? I am just focusing on the myoglobin. I don't want you to pay attention to the hemoglobin. Yeah. But here is our myoglobin. So come in and essentially it starts filling up and then it's harder. Okay, there's one thing I want you to keep in mind, that molecules are stupid, okay? All they do is they bounce around randomly, okay? They don't know, hey, I already bounced into a myoglobin that has oxygen, right? So it can't look for something. So now I want to take a trip to Crossgate Mall. And imagine the parking spots are myoglobin. So say we have 100 parking spots, 100 myoglobins. And the cars are the oxygens. And we have 100 oxygens. If you're the first one of the cars, you can find a spot pretty easy. You're parking right next to Old Navy, right? You're right there. That's where I park. And I have to go to the mall, which is not a fun thing for me to do with teenage girls. No. Okay, so the first one in, you're going to find a spot. Hey, the first 50. It's pretty easy. But what if you're like the 80th person? Is it harder to find a spot? If there's only 100 spots and you're the 80th person. But you're smart, right? You're not going to try to park on a spot that's already filled and crash, right? You're smart, right? You're going to look for an empty spot. So, but oxygens are not smart. They're going to go bouncing around until it finds an empty spot. So that's why you see this shape in this curve. This is why we don't just go straight up to 1. You see this curve because these last 20 out of the 100, so we're at number 80 oxygen, it's harder to find an empty myoglobin. So right here, maybe in your notes, up here, harder to find that empty myoglobin. Why doesn't it exactly get to 1? Because that last one is bouncing around a long time before it finds that one empty spot. Remember, it doesn't know. I've already been down this lane there, and I see that they're all filled. Okay, I know you guys do that, but oxygen molecules don't. I know that's really weird, but it works. Okay, so you guys got this shape? It goes up quick because there's a lot of empty spots. Then it slows down because we can't find as many empty spots. And they're stupid. All right. Now, hemoglobin has an S shape. Down here, it's different from the myoglobin, right? And up here, it looks more like it. Do you guys agree? That down at the bottom, a low oxygen concentration, it's harder. And then here, it's a lot easier. So I would say for 1 out of every 4 parking spots, you got to pay a toll for the hemoglobin. And then the 3 other people can come by. So the first one has to pay a toll, and then 3 other people can come in and bind easy. So that's, and what I say, and this is really on the molecular level, I call this hard binding, and I call this easy binding. So down at the bottom, it's hard binding. It's hard to bind. And then up at the top, it's easier binding. It's all easy binding with myoglobin. But here, on a molecular level, it takes more than one collision in order to get the oxygen to bind to the heme. Here, it might take just one, right? Because we've changed the conformation. Once we get the first one out of the 4 hemes in there, we're changing the conformation so then it becomes easier. So you can also put in your notes down here, this is the first one out of the 4. And then here, up at the top, we've changed that conformation to make it easier binding. So again, down here, hard binding. Up here, easier binding. It's hard binding because we haven't changed the conformation. The binding of the first one makes it easier for the other ones to bind. So we need more oxygen around, a higher concentration, in order to get the same amount of binding than we do with myoglobin. Yes?
Does the conformation change each time oxygen binds? Or
No, just once. Just once the first one's on. Then the conformation changes, and then the other 3 can come in. It doesn't change after the second and then after the third. No, just the once. Okay. Everybody good with this one? All right. This one is a totally different concept. I don't know why I decided to put them all in one slide, but just to make you write tiny. This is called the Bohr Effect. And this is why we can breathe. I want you to notice what happens to the binding or the amount of hard binding as we increase pH. This is all hemoglobin. Again, the axes are the same, percent saturation and the concentration of oxygen. What happens as we increase pH? Does it make it harder to bind or easier to bind? 7.6 is higher, so we get more binding. So let's look at 60%. We only need 20 millimeters of mercury to get 60% saturation, where at 6.8, we need almost 50 millimeters of mercury of oxygen in order to get 60% saturation. So we need less oxygen around at this higher pH to get oxygen in there than we do with the lower pH. So does it make it easier to bind with higher pH or harder to bind? Easier. It's easier to bind oxygen to hemoglobin at higher pHs. Another way to say this is that we reach the same percent saturation at a lower concentration of oxygen at higher pHs. Yep. We reach the same percent saturation of oxygen in our hemoglobin with lower concentrations of oxygen at higher pHs. I can say that again. We reach the same percent saturation, say 60, with lower concentrations of oxygen at higher pHs. So we only need a little bit of oxygen around at pH 7.6 to get to 60%, where we need 50% or 50 millimeters of oxygen at 6.8. And that's not that much of a difference of pH. That's a huge change. Yes? Sure. We need let's say, to reach the same percent saturation, we need a lower concentration of oxygen at a higher pH. That's what this plot is telling us. So therefore, we can infer that it is easier at higher pHs to bind oxygen to hemoglobin. This is not a huge change, even though it's a log scale, a huge change in the hydrogen ion concentration. But there's a very marked difference in that. This is what we call the Bohr effect. Same to do with the carbon dioxide as well. So if you have carbon dioxide and hydrogen ions around, you are going to drop oxygen. If you don't have hydrogen ions and carbon dioxide around, you're going to pick up oxygen. You don't have to write, to answer this question on the test, you don't have to write down this. I need to know that you understand that at higher pHs, as in the lungs, it is easier to bind oxygen to hemoglobin. So in the lungs, hemoglobin will bind oxygen and release our proton, as well as carbon dioxide. And we breathe it out. Breathe out our carbon dioxide. Protons are a product of metabolism, as we'll see later. And what happens is it makes working muscles have a very high hydrogen ion concentration. So since there's a high hydrogen ion concentration, those are going to go into the hemoglobin and kick out the oxygen. So then the oxygen's there for our actively metabolizing cells. The way we get rid of the protons in our lungs is to combine with bicarb, supposed to be a negatively charged on there, which makes carbonic acid, which we can break down to water and carbon dioxide and breathe out. Okay, you guys good with this? You've heard the Bohr effect before? Was it ever explained in this manner? No. So I think we go over a lot of the same ideas as you do in other classes. We just explain it in a different way. Hopefully not conflicting. Let me know if it conflicts with anything that you've learned before. Yes?
The bicarbonate is negatively charged?
Yeah, the bicarbonate here, HCO3, should have a negative charge. I think I tried to put it in there, but it meshes in with the arrow. See, I have it over here. but here I messed it up. So that combines to form carbonic acid, and then that can be breathed out. This is the same thing that happens in reverse in our oceans. This is why we're worried about the acidification of our oceans from greenhouse gases, because carbon dioxide will react with water to form carbonic acid, which releases protons and makes the ocean more acidic. Not to get political. The scientific protein folding. Many people's lives are dedicated to protein folding and studying this. There's graduate classes just on protein folding. Books and books and books, just talking about predicting how a protein folds. You can and you can't predict. I told you the story about tubulin and how the people came out with the X-ray, and we all whined saying that's not true because we had to have the solid to have the X-ray, and tubulin is such a big protein, as you can see here, that it needs to be in its aqueous environment in order to be native. So there's limitations to predict it, but there's a couple different things that we do. The bioinformatics is where we integrate computers with biochem. And I remember when I was in grad school a long time ago, this was a new field, and we had a postdoc that this was her job to try to figure out using bioinformatics what the tertiary structure of our tubulin was. And so what we do is we do a couple things where we are looking at something called sequence homology. So if you have, say, 25 to 35% the same amino acid sequence as another protein, you can hypothesize that maybe your protein folds similarly. And if you can't really find a similar structure, we try folding recognition. But again, hydrophobic interaction is the major factor in protein folding. So putting the nonpolar side chains inside, and the aqueous environment has the polar side chains around, is the driving force. And again, we're increasing disorder by having the hydrophobic interactions, the hydrophobic side chains inside. Because if we had them on the outside, we'd have that cage of water around them. And of course, I have a YouTube. I like this one. This is super cool. I could listen to this guy all day long, if it ever comes out.
So what we're looking at here is a result of the simulation of protein folding from folding at home. We're not looking at all of the atoms present because that would obscure what we're watching. So we're not showing it in the water, and we're not actually showing all of the atoms in the protein either. What we are looking at is mainly the protein backbone, which is that set of tubes that sort of go from red to blue. And we're also looking at the aromatic or a certain type of amino acid backbone, tryptophan, phenylalanine, and so on. The reason why is that the aromatic residues form what's called a hydrophobic core, and it's interesting to watch the core form. So what's going on is the protein's trying to assemble into its final state or fold. And it's basically getting some things right and some things wrong as it goes. And basically, as it gets something right, it doesn't, but not completely right, it ends up unfolding. Like right now, it unfolds and loses that structure, but then tries to come back into a state that's better. And so, for example, here it's finally reached the folded state, that the aromatic residues in red and green are basically in their final spot. the blue phenylalanine is flapping around, but that's the way it would be in nature. That part's not structured. It's basically reached the folded state and is perfectly happy to stay like this for a very, very long time.
That takes a microsecond. Is that awesome or what? What we just saw is what happens in nature for that protein to fold, and it takes a microsecond for that to happen. So it folds and unfolds and it folds again until it reaches its native state, and it's those hydrophobic interactions that's going to drive it. You're going to get those nonpolar, those aromatic side chains on the inside. Yes.
So was that imaging like made inside of a program, or was that just like a representation of something that they know exactly like how it like
I don't know how they made that video. I think they have some guesses about what happens during the folding of proteins. They might have seen things and other things that take longer. I don't know how they got that, but the guy sounded trustworthy.
Oh, yeah.
No, I think it was really neat. So just to drive that point home, the orange ones or the red are the hydrophobic side chains, and the green over here on the right are the hydrophilic ones. So you can see that the green are all on the outside, and the red are all on the inside. Now, when proteins don't fold correctly, bad things can happen. One of the things that we look for in biotech is what we call aggregate. When proteins aren't folded correctly, they clump up together, and you cannot inject somebody with an aggregate of a protein, of a biotech drug. So biotech drugs are proteins, and if you inject somebody with a misfolded protein, you will for sure get an immune response. It will drive their immune system crazy. So one of the things that my job was when I was in industry is to make sure that our product did not have any aggregate in it. And again, the prions, it can be toxic to the cell. You have misfolded proteins, that's what prions do. It will cause death. So, but on the other hand, sometimes we don't want proteins to be active until it's right at the right spot. So what we have are called chaperones that kind of stay with the protein until it's ready to fold. And then the chaperones aid in the correct and timely, or spatially, folding of the protein. Heat shock protein was the first chaperone protein to be discovered. I remember talking about this back before you guys were born. And chaperones exist in all our cells, from prokaryotes to humans. And this is another YouTube that shows how some of these work. Oh, really? It worked a couple months ago. This was so awesome. I'm sorry, guys. This worked. I checked all my YouTubes like just a couple months ago just to make sure that they would work, and it doesn't, I guess.
We forget.
You forget. Yeah. I try.
That happens.
Yeah. Yes.
So with the heat shock proteins, those are like where you get the cascade effects from sauna and stuff like that?
Oh, I don't know. I don't know. I'm not quite sure. I know the heat shock protein helps with folding of proteins. Beyond that, I'm not an expert with this one. But we can look that up. That would certainly be one of the things. So I wanted to know if heat shock protein was involved in heat shock. I'm guessing from the name it would be, but I know it's a chaperone. So unfortunately, try to find another website that will show this. But that is it. I'm going to give you guys a break.